welcome to this lesson. So in our previous lessons, we talked about what nutrients our body needs. In this lesson, we are going to be looking at some simple food tests that you will be conducting in your school lab for some of these nutrients. Now the nutrients we're going to look at will be simple sugars, for example glucose, for non-reducing sugars, which is, for example, sucrose. We're going to be looking at the test for starch, and for protein, for lipids, okay, and for fats. Alright, so these are the tests we are going to be looking at. Now, at the end of this lesson, you will see an activity 2. So activity 1 and activity 2 would be your assignment for this lesson. So, let's talk about simple sugars or reducing sugars and the test for those. So I put some links here. These are the links to the videos that we'll be looking at. Now I want you to actually see the color change and the colors of the solutions in these experiments. So the first test for the nutrients, simple sugars, for example glucose, we would use the Benedict's test. Now the method is to mash the food first and mix it with water. Now mashing the food increases surface area. And what that does is, instead of pouring your solution on one big chunk of food, we will mash it up so that we're breaking this up into smaller pieces so that the solution will be able to hit more places than just this one surface area. Alright, so basically 4 large areas. We've broken up into many little pieces. Okay, so that's why we mash the food first. Now some foods you might be able to see a color change immediately, but the best thing to do is to mash the food. So now we'll put each suspension into a test tube, and then you will add an equal amount of Benedict's solution. Now Benedict's solution is blue in color. Then we'll boil or heat the mixture in a water bath. Okay, the results will be, if there is glucose in that food, the color change will go from blue to green, orange to red. So it will go from blue to green and then orange to red. If there is no sugars, no glucose in that food, then there's no color change and the solution remains blue. Right, so let's look at a video of a demonstration of the actual experiment, and we'll pay attention to the color changes that happen. Alright, so here we have a stove to heat our test tubes. We have our Benedict solution, right? Our blue solution. We have some water. We have some mashed banana, cooked rice, crushed biscuit, and some salt. Right? And we have a dropper. Now we have a steel container with water because we are going to be using this steel container and water to place on the stove, and we'll put these test tubes in there to heat them. Okay, or to boil the solution. So let's take a look at this experiment. Okay, you can do this with 1 or 2 test tubes or any transparent glass, and you just wash them out each time. Right, but in your lab you will have more than 1 test tube to use, so you can do all the experiments at the same time. Now here he's putting the salt, the biscuit, the cooked rice. Again, everything is mashed. Mash the banana as well. Right, so he's adding some water. Now the water will help extract whatever nutrients we have in there. Okay, so he's adding the same amount of water and we're shaking. Shake well, that we extract any nutrient we have in there. Okay, now you're gonna make sure your hands are clean because we don't want any liquid from one test tube to go into the next test tube. Alright, now look at some color changing. Alright, then we're shaking again. Shaking. Okay, we're gonna observe the color. Now we're going to boil them. So we are putting them in something called a water bath. I think we spoke about water bath maybe in a previous lesson. Right? We heat the water and the water is transferred to the test tube and then to the liquid on the inside. Alright, the test tube. Alright. Now after the water boils, we make sure that the items in the test tube are boiling. Now some bad safety handling here, right? You're not supposed to be holding that with your bare hands. Okay. So observe the color change. Our salt contains no glucose, which is sugar. Right? Glucose is what we drink in Lucozade. And that's pure, simple sugars that go straight into your system, no need to break down. The biscuit is full of sugar. Now remember, we're looking for a blue to green and orange to red. Now the one that is most red here is this biscuit. So this biscuit is full of simple sugars. It's full of glucose. The salt has no sugars. It stayed blue. Now look at the cooked rice. The cooked rice stayed orange, right? So it has some simple sugars, alright? It still needs to digest into your system to get to that glucose. Now the banana has some sugars. It turned from blue to green. So there is some, but it's not simple sugars. It still needs to digest in your system to be able to extract that sugars in your body. Okay, so these are the color changes you're looking for with these tests. Alright, so let's look at the test for complex sugars or non-reducing sugars, for example, sucrose. Now, the name of this test is hydrolysis and neutralization, and then we do the Benedict's test. Alright, so we need to do a certain process before and then repeat the process that we just went through. So, the method is to mash the food again and mix with diluted hydrochloric acid. And then we allow that to cool. And then we add sodium hydroxide to the cooled hydrochloric acid and food mixture to neutralize the mixture. Okay? So later on when we learn about acids and bases and salts, we would know that hydrochloric acid plus sodium hydroxide neutralizes each other. Okay? And you will see fizzing happening. Alright, a simpler test for neutralization you could do in your own kitchen is you could take vinegar and baking soda, okay? And you would see fizzing happening, right? Between the baking soda and the vinegar. Because the vinegar is an acid and the baking soda is an alkali. Okay. So this reaction will cause fizzing. So that's a simple test you could do in your kitchen to show neutralization. Now when the fizzing stops, then you perform the Benedict's test. Now remember the Benedict's test is now we will already have mashed food that's mixed in some liquid, okay, which is the acid and the sodium hydroxide. So all we will do now is just add the Benedict solution, the blue Benedict solution, and observe the color change from blue to green and then orange to red. Okay, now if there's no sucrose in there, there's no color change. It remains blue. Alright, so there is no video because the video will be similar to the Benedict's test. Alright, the only difference is we're adding hydrochloric acid before and sodium hydroxide, and then you'll see fizzing. Alright, so let's do the test now for starch. Now, the name of the test for starch is called the iodine test. Now, this iodine test, you will do this in chemistry and in biology. So, in biology, we would focus on foods, yes, but we'll also focus on looking for starch in leaves of plants. So, to test for starch in leaves, you need to first boil the leaf to remove the green color, or the chlorophyll, from that leaf. And then you will do the iodine test to see the color change. Okay? Now, to do the iodine test, we're just going to mash the food and add a few drops of iodine solution. Now, if there is starch in that food, the color change will be from yellow-brown to blue-black. Okay? And if there is no starch in the food, it's going to remain yellow. So we have a video to watch for this one to look for the color change. Alright, look at the difference. Alright, so we see any color change that we're looking for. Alright, so iodine is usually a yellow-brown color. Alright, as you can see on the apple here, it's still yellow-brown. And then if there's the presence of starch, it turns black-blue or blue-black. Alright, so this is what we're looking for when we're doing this test. Alright, so now we're going to look at the test for protein. Alright, now the name of this test is called the Biuret or Biuret. Okay, it depends on if you're talking to an American or a British person, right? So, Biuret or Biuret. And what we do is we mash the food. Again, we explain the importance of mashing the food first is to expose a greater surface area, right? Now, some foods you can get away, just like the slice of potato before, where we just dropped the iodine test. That's because potatoes are full of starch. So it's easy for it to react with the iodine. Now, some foods you would need to mash up to have a greater surface area before you add the solution. Alright? Now, to test for protein, remember proteins mainly come from meat sources. So we're going to mash that food and mix it with water. Add a few drops of potassium hydroxide. Now, when we're doing chemistry later on, you will see how we come up with these letters here to represent these chemicals. Alright, so potassium hydroxide, or you could add sodium hydroxide to the food mixture, and then add a few drops of copper sulfate. And then if there is a positive result, if there is protein, the color will change from blue to lilac or to purple. Okay, now copper sulfate is naturally blue in color, so it will change from blue to lilac or purple. If not, alright, so if not, this is not iodine here, alright, the copper sulfate solution will remain blue. Okay, your copy of your notes will have the corrected versions there. Okay, so let's look at a video for the test for protein, and we're going to look specifically at the color changes and the types of foods that we're using in the experiment. So first we'll be experimenting on egg white, right, for protein from eggs, and then we'll look at presence of protein in bread, nuts, and in juice.
Egg white contains a lot of protein. The test for protein is called the Biuret test. This involves adding some sodium hydroxide solution, then running some very dilute copper sulfate solution down the side of the tube. At the top of the mixture, The colour change to mauve shows that protein
So we're
is
seeing
present.
that lilac or that purple
So
color.
which of these will test positive for protein? Bread in tube 1, nuts in 2, and grape juice in 3.
Alright, so this is a nut. The bread, sorry. You have a color change. This is a
Both
nut.
the bread and nuts contain protein. Which contains more?
Okay, so the question is which contains more? Which one is more purple? The nuts, right? So bread does have some protein in it. Alright, there's a slight color change here to lilac, but the nuts contain more protein. Right? So if you don't eat meat, the best substitute for meat to get your protein from are nuts. Okay? And there is no presence of protein in the grape juice. Now the juice, if you go back to in the video, you will see that the juice is originally this color. So now we're going to look at the test for lipids, right, which are fats. And we have two tests. We could either do the grease test and we could do the emulsion test. Now, the grease test would just be to take the food sample and we're going to rub it onto some filter paper. Now, filter paper is something in the lab that they use to separate solids from liquids. And we're going to rub the food sample on the filter paper and we will see, we're going to leave it to dry to remove any water that was in the food. And when you hold the filter paper up towards the light, you are going to see, right, a greasy translucent spot on paper. A translucent meaning you could see through partially, right? It's not transparent where you could see through clear like glass. It's going to be translucent. If there is no fats, there will be no greasy spot. Now the emulsion test, this way, we would need to first mash the food, dissolve it in ethanol, so ethanol is a type of alcohol, and the lipids are fats, so it will not dissolve in water, okay, so but it will dissolve in ethanol instead, right? So that's a type of fat like oils. If you throw oil in water and you shake it up, the oil will not dissolve. Okay, but if you throw the oil into some sort of alcohol, you could maybe try it with rubbing alcohol, it will dissolve. And then you will pour the solution into a clean test tube of water. Now, if there are fats or lipids, we will see a white cloudy emulsion being formed. Right? If there are no fats, the solution does not turn cloudy and white. Okay, and we will look at a test for the presence of lipids or fats. Alright, so let's do the ethanol emulsion test for fats. And we're going to look at a short video and remember to look for that white cloudy solution in the video. Okay. Alright, so we dissolve the fats into the ethanol and then we pour the solution into a clean test tube of water, or you could add the water to the test tube as she did. Alright, which would be the better thing to do. So we saw that white cloudy emulsion being formed. That oil is full of fats. Okay, you could take the oil and you could try to test with the other solutions, like the Benedict solution, and look for glucose, right? And see if there's any color change. But we know that oils are full of fats, and this is the positive solution that we want to see from this lab. Right? Now, let's talk about your activities. I'm going to talk about activity 2 first. So, what you need to do is to go through all of the notes that we just did. It doesn't matter what order you choose. Okay? So, let's use this one here. Let's use the nutrient. The nutrient is going to be fat. You're going to put in fats here. Fats or lipids. Okay? The name of the test. Now, fats or lipids have 2 tests. So, I want you to do both tests. Okay? Name of the test, you're going to start with 1, the grease test. Okay? And then in the next column, you're going to put 2, the emulsion test. Alright, so you redraw this table on your own paper and fill in all the tests that we went through just now. Your reagents for the grease test. All you had was a piece of food, right, and filter paper. So the food and the filter paper. Right, in your procedure, you're going to list what you need to do, your steps. If there's a positive result, for example, the grease test, you would see a translucent spot. Okay, so you write translucent spot seen. Negative result, no translucent spot seen. For the emulsion test, you put in your reagents, which is going to be your fat or your food. Okay, we don't know if you have fat yet, so your food, right, and your reagents that you need, including your water. You list your procedure. You say what positive result you're looking for, white cloudy emulsion. Negative result, solution does not turn cloudy white. So you're going to fill out this table for me with all of the information from all of the tests from before. Alright, now you need to memorize that table. Okay, so this is one of the activities that is going to be your assignment. Now, your activity 1 is you're going to do the same thing. For activity 1, you're going to use the simulation. There's a link in the classroom, and you're going to complete this table. So I'm going to show you now what that simulation is and how to use it. Okay, so in your folder classroom, you will see the simulation link. If you hover over the link, you will see simulation. And you are going to be using this for detection of starch in food samples. Alright, so it's the iodine test. Now, how to use this? You will click on your food item here. For example, let's start with the carrot. And here's your carrot extract. And you will drag your dropper there. Okay, so you put some drops and then drag the dropper back into the bottle. And you're seeing there is no color change. Alright, remember what you're looking for the presence of starch, right? We need this brown yellow solution to turn blue black. So there is no color change here. So there is no. Alright, now you will go back to your table. Alright, carrot, is starch present? No. Okay, well, you don't need to write that. Alright, if starch present? No, starch is not present. So you tick this column. Alright, so let me do the other example, the wheat for you. So when you need to do the test for the other one, you're going to click on wheat. Right, there's some wheat appeared on your plate. And you're just going to drag that dropper onto the wheat. And you notice an immediate color change. Alright, so change from this color to blue black. Yes. Okay. And you're going to come back to your table and you're going to click yes, starch is present. Okay, so you continue your simulation and do it for the apple, the egg, tomato, oats, potato, and bread. Right, and fill out your table. And for your assignment, you're going to submit this table, completed table, as well as the completed activity 2 table. Okay. And that will be the end of today's lesson for food tests. And these food tests are very important. If you're doing integrated science later on, you need to memorize these food tests. You need to memorize the name of the test, the reagents, the procedure, and what you're looking for positive or negative results. Right? If you're doing biology, the same thing. Okay. Alright, so complete those 2 activities, submit them, and I will see you in the next lesson.